In this lesson, we're now talking about pathogenic molecules and their virulence factors, so specifically viruses and prions, as opposed to bacteria, fungi, protists, and parasites that are living organisms. So we need to distinguish between those two. Okay, these are molecular pathogens, and these are diseases caused by pathogens that are not cellular. Okay, cellular, we're talking about consisting of, it could be a single cell, it could be multiple cells, it's got cellular machinery, it's got organelles, it's got all those things that are capable of reproducing. However, non-cellular pathogens are really not capable of doing that. They need to use our cells in order to reproduce, so they are not living organisms. Now we're going to talk a lot about antigens. Antigens are specific molecules that which the host cell can recognize as foreign and will respond to. So they can be found on the cell surface of a normal host cell, bacterial cells, so our body knows, hey, that's not us, that's some kind of foreign material and I need to get rid of it. All right, non-cellular pathogens are generally smaller than, say, unicellular pathogens. We're talking really quite small here. These are viruses here, as opposed to, say, bacterium, which are here. And if we have a look, this is actually a log scale. So it's not, you know, this is only a teeny tiny bit smaller. It's quite a lot smaller. Prions, which are essentially a type of protein, they're the smallest known pathogens. And we'll talk about them next time. Okay, we're going to go through the same format exactly. We've still got the pathogen, but we're talking about the features, the transmission, the virulence factors, and some examples. Okay, now viruses are obligate intracellular pathogens, which means that in order for them to do their job, they have to be intracellular and they have to get into the host cell tissue, basically. And they can vary substantially depending on the virus family, but they all have basically the same structure. They're containing some kind of genetic material encapsulated in some kind of protein outer shell. They're all carrying DNA or RNA, so it's, you know, a viral genome. And it's going to encode for instructions that make new viral particles, and they come in all wonderful shapes and sizes. Their nucleic acid is surrounded by a protein capsid, and many of them also have these little envelope proteins as well to help them evade the immune system. So once inside the host cell, the viruses actually have to survive our own cellular defenses, and our cellular defenses include things like having enzymes that can break up foreign DNA and RNA. So really, once they've injected their nucleic acids into the host cell, they have to be very, very efficient. And it's going to instruct that cell to make copies of itself, so that eventually, when it's got heaps and heaps of photocopies, it's going to burst the cell, they're going to be lysed out and go and spread and go and infect further cells. So it's a pretty efficient system on their behalf. Now, viruses are really, really specific to their host cell or organism. It's got to infect that particular cell, so it's really got to be specific to the receptors on the outside of the cell membrane there. So, for example, the adenovirus infects epithelial cells in the upper respiratory tract and basically gives us, you know, a common cold. Now, viruses can spread through so many different ways, right? Direct contact, indirect contact. Hey, you know, again, I've sneezed on the desk and I've rubbed my hand on it, or someone sick is using my keyboard and I've managed to touch the keyboard and then rub my eyes, whatever. They can be in droplets that are in the air. They can stay airborne for a very long time. We're talking about fecal oral transmission, so you're not washing your hands when you go to the bathroom, and vectors as well. So we can be talking about so many different things, unfortunately for us, but it's a pretty effective way of spreading themselves around as a virus. So there's millions of different viruses. They vary greatly depending on the structure in their, you know, they have virus families essentially, and they're going to vary in terms of what organism they're capable of infecting, and vary greatly in how rapidly they're able to change and mutate. If we look at, for example, influenza, you know, the likely transmission here is aerosol droplets in the air, contact with contaminated objects, direct contacts with infected individuals. We kind of have that understanding, and it works in terms of an annual cycle. We know that when it becomes winter, it's cold, people are getting sicker. Every single year, those numbers will go up and down. Again, the replication is exactly the same. It's going to get into our host cells. It's going to use our genetic processes to multiply themselves, and then they're going to be released to go and infect other cells. Dengue fever is an interesting one, and it's a vector transmission. So it's again kind of using those mosquitoes to work around the equatorial regions in tropical areas. It's a pretty serious case of the flu, but has a very, very deadly outcome. And I've got some really interesting stories about a university professor that I had in a long time ago who would inject himself with it to see what it was like. And most people who have dengue fever can recover, but sometimes it's pretty serious and can cause a lot of fatalities. And if we're talking about things like climate change, as the warmer tropical weather moves further down, say Queensland, we start to see more and more cases of it. Now, viruses, in terms of their virulence factors, they are attached to host cells. We know that that's really important to them. They have to have some kind of spiky protein, whatever, on the outside of their cells so that they can get in. So they don't just have to invade, they have to evade as well. So they have to invade the actual cell and get in, then they have to evade our host immune system. So there's lots of different ways, and they can have an envelope around them that's derived from the cell membrane of the host. So it actually, they're taking our mechanisms and going, hey, you can't see me, look, I've got this invisibility cloak on, which is really, really quite interesting. But it also does allow them to change and mutate, basically, to evade our systems for even longer. Now, this ability to evolve their genome as well is by reshuffling what they have is a problem for us. So for example, we need a new flu shot every single year because that flu virus changes. So it's not that, hey, I got that last year, I'll do it again. It's like, no, there's probably a new strain because all of that genetic information has rearranged. It's probably got some new horrible superpowers that mean that maybe their invasion factors have increased their pathogenesis. In terms of influenza, like, again, it's pretty clever. It's got membrane attachment and fusion factors in this hemagglutinin, and this neuraminidase is a surface protein that allows the virus particles to be released from that host cell. So that's really needed for that viral replication. They have amazing adaptations for them, but terrifying for us. And here is a delightful list of viruses that are pretty commonly known, and you can see that the mode of transmission is incredibly variable depending on what it is, and there are really large classes of different viruses. Specific to viruses on this one, prions in the next lesson.